Good afternoon, everyone. Welcome. Glad to see so many of you here. And there are a lot of people on Zoom as well. And it's a great pleasure to welcome our visiting professor and special guest today, Dr. Clifford Rosen. He's a basic and translational bone biologist. He's the principal investigator of the Northern New England Clinical and Translational Research Network that stresses clinical translation of basic investigations. And he's a very good example of somebody who has done this very successfully for a long time. And he has served 7 years as chair of the External Advisory Committee for the Mayo CTSI, and previously served on the IOM committee on the CTSA program. He currently oversees the Rosen Musculoskeletal Laboratory at the Maine Medical Center and is an endocrinologist. Dr. Rosen has 30 years of continuous NIH funding. In the last 10 years, his lab has been studying mesenchymal stem cell fate, in particular reference to the switch between pre-adipocytes and pre-osteoblasts, and focusing on the bioenergetic programs of those progenitors. There are currently 4 NIH-funded projects in his lab focusing on this theme of lineage allocation in its biochemical determinants and alterations in osteoporosis. Dr. Rosen is the director of the Physiology Core of the P20 COBRE, co-director of the Mesenchymal and Neural Regulation of Metabolic Networks, NIGMS. Most recently, he is the multi-PI for the ISCORE network of NIH-funded idea states doing the long COVID study RECOVER. So Dr. Cohen and I have the pleasure of seeing him on Zoom almost Tuesday afternoon? Tuesday afternoons. It's a great event in my week. He has served a 4-year term at the NIMS Council, board member of the Endocrine Society. Not listed here, he was on the American Board of Internal Medicine Endocrine Section jointly with me, where I got to know him very well at that time. He has published more than 660 publications in journals like Nature, Nature Medicine, Cell Metabolism, PNAS, New England Journal, Journal of Clinical Investigation, and The Lancet, all the top journals. And he's been associate editor of the New England Journal of Medicine for 11 years, and I'm sure we've all had papers rejected by him.
Including me, too. Thanks, Vivian.
It's really a pleasure to welcome him here, and he's going to talk about the evolution of parathyroid hormones.
Great. Thank you. Thank you. It's great to be here. And Vivian and I are old friends. We go back a long, long time, and it's so great to see him. And he's done really well. And his editorship at Diabetes Care was hugely successful in raising the impact factor and success of that journal. So what I'm going to do today is tell you about something that may not be in your present day life every single day. It is where I live up in Maine. But the idea in this talk is to give you sort of an update about parathyroid hormone and some of the newer work we're doing. And it's really not new. It dates back 100 years when some scientists first discovered that if you give parathyroid hormone, you could build bone. But about 50 years ago, my old colleague and friend, a Southerner, Norman Bell from South Carolina. I don't know if you knew Norman, but anyways, he made a very seminal observation in the journal Metabolism that was buried for 45 years. And I'll come back to it during the course of my talk. And then more recently, work by Bruce Spiegelman, who has described a role for PTH that among boneheads such as myself, people who work in the bone field were really skeptical that parathyroid hormone did anything other than regulate calcium homeostasis. So what I'm going to do is going the wrong way, provide you with a brief overview of PTH and then talk about cold exposure because that's been a big part of this winter in Maine. And it actually led us to some clues as to the physiology of parathyroid hormone. And I think, the bottom line is science is very exciting. You have to follow the science to help you really explore new avenues of physiology and metabolic health. And so I hope I'll be able to show you how we've done that and what work we need to do as we move along. So the National Weather Service published a seasonal outlook in November last year, 2025. And they predicted that there would be about average temperature and precipitation for New England. As it turns out, this is a picture of my driveway, the average temperature departure was 5 degrees. That's the mean temperature departure. That's huge. We went about 40 days without ever reaching 32 degrees centigrade, and we had a ton of snow. So we were shaken to the bone. An old term that was used when people were in crisis situations. And I'm going to come back to bone because it's a major target of cold exposure. And then I'm going to try to tell you how parathyroid hormone works in that process. So many of you know that brown adipose tissue is an important mechanism to generate heat. We can generate heat through shivering, which we did a lot of this winter. And tonight we're supposed to get 3 more inches of snow and come back from this beautiful weather. So I have to adapt again. So I need to have some of my brown fat generate non-shivering thermogenesis, which is efficient. And with PET-CT scan, we can visualize brown adipose tissue. Its function is not just to generate thermogenesis and active heat, but also as a sump for glucose and fatty acids. And so higher mass of brown adipose tissue has been associated with improved cardiovascular function and lower the risk of diabetes. So brown adipose tissue is activated by adrenergic drive. And so during cold exposure, TRPV cold sensors actually drive the hypothalamus to release signals to enhance sympathetic output. And that sympathetic output goes to brown fat, where it's highly innervated, but it also goes to other tissues, including bone. So to understand the potential detrimental effects of being shaken to the bone by cold, I just want to review for you very briefly the bone remodeling unit, because every 10 years we get a new skeleton, and this remodeling process is a function of both dissolving old bone and adding back new bone. And as we age, we are able to couple those 2 units, bone forming cells, osteoblasts, with bone resorbing cells, osteoclasts. And we do that because the osteocyte, the embedded cells in the bone matrix, are able to send signals that generate both osteoblasts and osteoclasts to work. So the osteoblasts are forming new bone and the osteoclasts are resorbing bone. And this sort of symphonic process keeps our skeleton exactly where it should be in terms of maintaining bone mass. So what happens with cold exposure? Well, we know that we published this about 15 years ago, that if you put a mouse in 22 degrees or 23 degrees, which is room temperature, and compare it to a mouse at 30 degrees or 32 degrees, which is really thermoneutrality for the mouse, the bone density is much lower. So you have thinning of the trabeculi and thinner cortices. And if you take away brown adipose tissue, you actually make that bone thinner. And in addition, Dew and colleagues showed that if you transplant back brown adipose tissue, you actually can preserve the skeleton in a cold exposed state. So the first clues are that brown adipose tissue has a role in in regulating bone density. And in the cold exposed animal, you get this total uncoupling of bone resorption from bone formation. So there are less cells that are forming new bone, and there's a marked increase in the breakdown of bone, what we call increased bone resorption. So this uncoupling is almost certainly the mechanism for cold-induced bone loss. And we know from ecological studies that individuals who live in higher northern latitudes, so you guys are not at risk, but those individuals at higher northern latitudes have a greater risk of fracture, hip fractures. And this is particularly true in older individuals. Now, people have previously subscribed that to vitamin D deficiency, but we see that that in these populations, many of these are vitamin D sufficient, and they still have a greater risk. So it's possible that the long-term cold exposure has a persistent issue. The other thing we know is that changing climates from a warm climate to a cold climate. So if you were to come to Maine now, you would activate your brown fat, but you would also show this increase in adrenergic drive, which leads to increased bone resorption. So it's not just chronic metabolic adaptation, but it's acute metabolic adaptation as well that's important. And we know, and we've published this previously, that brown adipose tissue mass, the volume of brown adipose tissue mass, is directly related to bone density. So immediately we have this fat-bone connection. And what I want to tell you is that that is one clue as to how parathyroid hormone may work beyond calcium metabolism. So stay tuned, because I'll just reemphasize that cold exposure increases bone loss, probably by increasing bone resorption and uncoupling the remodeling unit. Brown adipose tissue plays a role in modulating this effect, or at least is related to bone density. So the question that we've been asking for a while is, how does the human skeleton protect itself from long-term cold exposure in humans? Why don't all of us that live in Maine just have higher bone resorption and lose their bone mass at a higher rate? What is it about our physiology that allows us to protect our skeleton from acute and chronic cold exposure? So I'll switch gears, because I want to talk about parathyroid hormone for a minute and tell you that when we evolved from fish to mammals, one of the most important protective mechanisms as you get out from marine life, which has a lot of calcium and salt, to terrestrial life, is a need to maintain calcium balance. And that occurred by the evolution of parathyroid hormone, the parathyroid hormone receptor, which is present in fish, and we'll come back to that a little later, all the way up to the ligand, which is parathyroid hormone or parathyroid-related peptide. And so this evolution guaranteed that we could maintain calcium balance, and parathyroid hormone is essential for that function by stimulating bone formation and also upregulating bone resorption to maintain this coupling phenomena. So with calcium-restricted diets, the first thing that happens is this upregulation of parathyroid hormone. And PTH can then drive osteoclasts to resorb bone so that the balance of calcium is absolutely maintained. And it's this evolution of parathyroid hormone and vitamin D, which is the active component of vitamin D125, is directly regulated by parathyroid hormone. And that's how we got to where we are, that we didn't need an environment that was calcium rich in order to maintain our skeletons. So parathyroid hormone works through the parathyroid receptor, or PTH1R, and it works in the kidney, it works in the gut, and it works in bone. And in those three areas, in the gut, it increases 125 vitamin D. In bone, it increases bone resorption with direct action on osteoblasts to tell the osteoclast to digest bone, and in the kidney to save calcium. But also has a very profound effect on stromal cells in the bone marrow. And one of its roles is to stimulate new osteoblasts from stromal cells and increase bone formation. And this has been well described by a number of investigators over a long period of time. When you give intermittent parathyroid hormone, that is, you give a shot, it lasts about 4 hours and goes away. You can trace that increase in the maturation of these skeletal stem cells to become osteoblasts. And some of those osteoblasts then age and get embedded within the bone itself and become osteocytes, which are regulatory components of that remodeling unit. So we and others have shown multiple times that parathyroid hormone can increase bone formation, and it's relatively dramatic. And this is one of the reasons why parathyroid hormone, or Forteo, has been used for the last 25 years to build bone mass. So when we prescribe parathyroid hormone, we give it once a day, and it lasts about 4 to 6 hours. You get an increase in bone formation, you get an increase in bone mass. Ultimately, over time, that increase levels off. So you can't continue to use it for long periods. Generally, it's prescribed for 2 years. And it's got a great safety profile, and it really has been the first of the anabolic agents in the treatment of osteoporosis. Anabolic agents meaning ones that directly stimulate new bone formation. The previous therapies, including Fosamax and Actonel and the other anti-resorptive drugs, work by blocking bone resorption, but parathyroid hormone stimulates new bone formation. So far I've told you the very classic approach to parathyroid hormone. It's a regulatory feature of calcium metabolism. It's essential to get calcium out of bone rapidly. And it also can be used to stimulate new bone formation by increasing the recruitment of osteoblast progenitors. And importantly, fracture risk reduction is quite dramatic, 50 to 60% reduction in non-vertebral fractures. So this drug has been now out for 25 years, and the risk profile is really quite low. It's very well tolerated. There's no hypercalcemia, and the individuals can build 5 to 10% of their bone mass back after treatment. Okay, so we were interested in parathyroid hormone and any other of its features that might be evident. This is really something that we've been working at in the lab for some time, and that is trying to understand what parathyroid hormone does to these stem cells. And one of the reasons we were interested is because we know that these early progenitor cells that I showed you, if they don't differentiate into osteoblasts, they become adipocytes or fat cells. And so we wanted to try to understand the mechanism behind that switch. And the reason we did is because we had data that with aging and with osteoporosis and with other conditions, the bone marrow begins to expand its fat cell population. And so we wanted to know, is parathyroid hormone regulating that switch between fat cells and bone cells? Because the more fat cells you have in your marrow, the lower your bone density. So we were intrigued by this. And so we took a very direct animal model approach. We deleted the parathyroid hormone receptor, And we did it in the early progenitor cells. So way back here, we decided using a promoter to delete parathyroid hormone receptor, which is highly expressed in those early progenitors. And what we found was no big surprise. Bones were terrible. The lack of parathyroid hormone during development led to these really almost non-discernible bones. These were bones that fractured easily, and this was only at 6 weeks after we deleted the parathyroid hormone receptor. So that wasn't a big surprise. The mice, we had to feed manually because they couldn't put their paws down because they were so poor in their bone mass that they had to be fed on the floor of the cage. So this was very destructive to bone and not anything earth-shaking. But in the process, we found that there was tons of marrow fat in the bones. So the bones were weak, they were structurally impaired, functionally impaired, and they had tons of marrow fat. And this led us to try to understand what was the mechanism that PTH regulated that would prevent this with the administration of parathyroid hormone. And we identified a gene, zinc finger protein 767, which has a whole separate story that I'm not going to talk about today, as being one of the major modulators of that shift. So parathyroid hormone regulates bone marrow stromal cell lineage fate, and it's an important one. So as I mentioned, marrow fat is a vital part of the bone marrow. For many years, people thought marrow fat really didn't do anything. It was a filler. So as we age, our hematopoietic system slows down, our bone forming system slows down, so fat comes in and just takes over that space. But in fact, we now know that that's not the case, that these adipocytes are unique. They're of special origin because they come from the bone marrow, not from the peripheral fat, and they have very, very different functional implications. So with the idea that parathyroid hormone prevented that fat and build bone, we really got interested in trying to understand the mechanism of that. So to do that, we turned to a human model of calorie restriction, anorexia nervosa. And in anorexia, individuals have a very high fracture risk, and they have a lot of marrow fat in their bone marrow. In fact, normally bone marrow fat is about one-tenth of the total fat mass in our bodies. But with anorexia nervosa, about a third of the total fat mass is located in the marrow. So we were trying to understand, so what is happening? Why is marrow fat in the bone marrow? What's it doing there? And can we do anything about it? And is it detrimental to, because we know the skeleton is weaker with anorexic women, and they fracture much more readily. So we couldn't use that model in our labs, so we switched to a calorie restriction model where we reduced the amount of calories that a mouse would get by 30%. And they lose weight, they lose about 25% of their weight. Their bones thin, and their bone marrow fat expands exponentially. So you get, within 1 week, you get this huge increase in bone marrow fat. And this allowed us to ask an even more important question, is that what is that marrow fat doing in the skeleton? So the first thing we did is treat those mice with parathyroid hormone. And what our strategy was, but it was probably not the right strategy, was maybe we can make those fat cells not become fat cells, but become bone cells. And so we could trick the body into switching the fate of those stem cells into bone cells. And sure enough, we showed that bone marrow fat was markedly decreased by parathyroid hormone. But we were wrong in thinking that it was all related to that lineage switch. In fact, that was only a small component of it. And so this led us to ask the question, what role does parathyroid hormone play in the balance of energy metabolism within the system. So what we found was when we look carefully at the bone marrow fat, the size of the fat cell was markedly reduced. The number of fat cells wasn't changed, but the size was reduced. And that led us to believe that potentially what parathyroid hormone was doing was causing lipolysis of the fat cells in the bone marrow. And why would it do that? Well, it might do that because it might then provide fatty acids for energy for the osteoblast. So we previously, most of us thought that bone marrow fat was bad for you because it was releasing cytokines and obesity in general is bad for you. But what we learned is that the bone marrow fat is a compensatory mechanism in the body for the lack of substrate or energy. And by stimulating lipolysis, you could actually release energy stores within the bone marrow that could help form new bone. So that led us to try to understand what the role of parathyroid hormone is in overall metabolism, independent of calcium. So put aside everything we know about parathyroid hormone as a metabolic determinant of calcium balance, and put aside the fact that PTH can increase bone mass by changing stromal allocation, and ask the question, what role could parathyroid hormone play in overall energy metabolism? So Norman Bell published in 1976 in Metabolism a seminal paper entitled On the Lipolytic Action of Parathyroid Hormone in Man. 1976, March of 1976, exactly 50 years ago. And in there, he did just very simple experiments. But you know, in science, you never come up with anything that nobody's thought about. There's always something that people have done in the past. It's just finding that data somewhere. So anyways, who would believe this bonehead, Norman Bell, who did this one experiment and one paper in which he gave bovine PTH and he measured glycerol production in fat tissue from 3 patients. And 8 normal subjects, 3 patients with hypoparathyroidism, and 8 patients with pseudo-hypoparathyroidism. And what he concluded was PTH, because it works through cyclic AMP, can induce lipolysis in peripheral fat tissue. 50 years later, we showed that if you give parathyroid hormone to hypoparathyroid individuals who don't have any PTH, not only do you improve the calcium homeostasis, but you also reduce the bone marrow fat significantly. So this observation was rather dramatic, and it led us to think that maybe parathyroid hormone's actions were fairly significant in fat tissue. So then we went ahead and published a few years ago where we took the bone marrow adipocyte and actually were able to show that that with the release of fatty acids, that you could build bone, that the osteoblast actually needed those fatty acids to make bone. And so the idea that fat mass alone is detrimental to the skeleton has been revisited by us and others with the idea that these fat cells show up, particularly under times of stress, and potentially are mobilizable by different hormones and compounds. And so science is so exciting and so interesting that it never ends. So we were sure now that fatty acids were the source of energy for osteoblasts that were struggling. So you have a bone population and they're struggling because they're under stress, either because they don't have enough energy or they have osteoporosis or they have other things. And here you go, you give parathyroid hormone and you get fatty acid release and you get some energy improvement in the cellular model using bioenergetics. So that was great until about 3 years ago. Lawrence Kazak, who trained with Bruce Spiegelman, called me up and he said, Cliff, he said, I know you are thinking about fatty acids, but have you ever thought about glycerol as being the other compound that has an impact on the bone marrow, and in particular on osteoblasts? And I said, oh, no, glycerol doesn't do anything. You know, we've done studies. When we culture bone cells progenitors, we do add beta-glycerol phosphate, but you know, it's because of the phosphate. It's not because of the glycerol. Well, long story short, 3 years later, coming out in Nature next month, we have a paper with Lawrence showing that glycerol, the other released factor from fatty acid, from adipocyte lipolysis, that glycerol is actually a critical element in activating tissue nonspecific alkaline phosphatase, or ALP, alkaline phosphatase, which is absolutely essential for mineralization. So not only do the fat cells release fatty acids that help fuel the osteoblast work, but they're also releasing glycerol, which activates extracellular TNAP and begins the mineralization process. Because osteoblasts, to form new bone, they have to lay down collagen, and then they have to mineralize that collagen to make it sustained. So anyways, it's a side story, but it's an interesting story because you never can say this is absolutely the only thing that happens in science. There's always other alternatives. And then Ryan Riddle in a competing group at Tom Clemens' lab showed that not only does PTH mobilize fatty acids from the bone marrow, but also from peripheral fat cells. So we're now getting a clue that PTH has effects other than just what it does in the bone marrow. Okay, so big deal. Does that make any clinical sense? Well, it turns out we went back and looked at some parathyroid hormone studies for osteoporosis. And wouldn't it be the case, but 90% of the papers that publish on parathyroid hormone and bone do not even present weight data. They do not show whether people who are getting PTH might lose weight. And so we finally got a collection of data that shows that there's a modest weight loss with parathyroid hormone injections for osteoporosis. So that was kind of interesting. And then in a mouse model of hyperparathyroidism, you can see that same loss of fat mass. So there are clues now that PTH has some independent role. So what we did next is, thanks to some reviewer comments from one of our grants on R21, they said, well, you haven't shown that PTH actually does anything in vivo. So we designed an experiment giving a standard dose of PTH daily for 5 days to black 6 mice. And what we showed is that particularly in males, this marked reduction in peripheral fat tissue. So suggesting that, in fact, we can replicate that in vivo lipolysis. But that wasn't satisfactory enough for reviewers, so we went ahead and did metabolic chamber work. And what we showed was that PTH dramatically stimulates energy expenditure in animals that are given PTH for 5 weeks. And that effect is, interestingly enough, more on the daytime energy expenditure than on night. In addition to that, we cold exposed mice that were treated with PTH versus those that were not, and those that were exposed at room temperature. And the parathyroid hormone treated animals were protected against a marked decrease in their core temperature. So it gave us clues that PTH could, in fact, have an impact. And we also showed that when we took the brown fat out of the PTH mice, that in fact UCP1 is markedly upregulated by PTH, and ZFP467, that molecule that we had isolated from the bone marrow stromal cells, was markedly suppressed. So we think 467 is a negative regulator of brown fat energetics, and that suppression is necessary to allow an active thermogenic process. So more recently, we looked at proteomics and asked the question, if you give PTH, what happens in the circulation? Are there things that come up that are unique to parathyroid hormone treatment? And one of them was RBP4, which was markedly upregulated. But what comes up consistently is lipid mobilization. So even though we are fixated on parathyroid hormone as a calcium metabolic pathway, obviously the clues are there that this is an important component of lipid lipolysis and lipid mobilization. So we then asked the question, if this affects brown fat, what is the mechanism and what is the target cell? So we had some clues. I did a sabbatical in Bruce Spiegelman's lab 2 years ago for 6 months, and it was really fun and fantastic. And he walked by me one day. He says, "I know why you like it so much, because you're not in your own lab." And I guess that's what's great about a sabbatical, is that you can be removed from the immediacy of running a lab. But anyways, Bruce reported, and most people in the bone field thought the paper should never have been published because they could not believe that parathyroid hormone or parathyroid-related protein could actually induce browning of adipose tissue, particularly in the inguinal depot. And he showed not only that PTH could induce a thermogenic program, or PTHRP, but that it also led to muscle cachexia and was a component of the cancer syndrome of wasting that occurs with high levels of parathyroid hormone-related protein. So there was a template for an independent effect of PTH on fat tissue. But Bruce went on to other bigger and better things and really didn't follow up the story. So we had a sense that there was probably more to it than that. So brown adipose tissue is really a mixture of many different cell types. There's neurons, there's brown adipocyte progenitors, there's vascular smooth muscle cells. There's a whole bunch of different cell types. And although Bruce was able to show in vivo that PTH induced a thermogenic effect in inguinal tissue, we were interested in trying to understand what the target was in brown adipose tissue. So we had evidence that it could induce thermogenesis PTH, that it protected mice from the cold with long-term therapy, but we didn't quite know what the mechanism was for that. So we first wanted to identify the cell type. And I had to lean on one of our young former scholars who came from Joslin, Matt Lines, who was in Yeehaw's laboratory at the Joslin with Ron Kahn. And Matt came to our group and was very interested in following up his Nature Metabolism paper, which suggested that TRPV1 positive progenitors that were originating from vascular smooth muscle cells were the subsequent progenitors of adipocytes that respond to cold. And so this was his initial observations. And so he began a data mining study with us where we looked through all the single cell studies on brown adipose tissue in mice. And we were looking particularly for the PTH receptor. So, you know, Bruce had shown that it worked in inguinal tissue. There really wasn't a lot of work on what the PTH receptor expression was in brown fat. And so this confirmed Bruce's findings that in inguinal white adipose tissue, the PTH1R was co-expressed with TRPV1. So then we went and looked in these studies of mouse brown adipose tissue and found the same thing was true, that PTH1R was co-expressed with TRPV1. And in 7 studies of single-cell brown adipose tissues, we were able to, using data mining, to identify that PTH1R was associated with TRPV1, but also that ZFP467, our protein that we had isolated from bone marrow cells, was inversely related to UCP1. So as UCP1 went up, ZFP467 was suppressed. That confirmed that this is one, ZFP467 is a modulator of thermogenesis. So Matt's pretty creative, and he wanted to understand in his lab the function of brown adipose tissue. He had published some work on 1213 dihome, which is a lipid product which is produced from brown adipose tissue that improves insulin sensitivity. So he wanted to create a model in mice that looked at the role of exosomes in messaging brown adipose tissue to different peripheral tissues. So we now have begun to appreciate that brown adipose tissue is an endocrine organ, that it's secreting what we call cytokines. It's also producing fatty acids that can signal to fat tissue, other peripheral fat tissue, can signal to the pancreas in particular to enhance insulin secretion. And so we built this GFP model in which the cells in the animals that express TRPV1 will show green fluorescent protein. And again, his target was trying to understand whether exosomes were one product of brown adipose tissue. And so using CD63 as a label with GFP, you could pretty much confirm that what's being produced are exosomes. And these are small molecules, vesicles that come out of the membrane and carry cargo, which may have distant effects. So it's an advanced form of endocrinology with smaller molecules that have distant target effects. So initially, he showed that TRPV1 positive cells are present in brown adipose tissue. And surprisingly, in the marrow too. And that really intrigued us. So he called us up and said, Cliff, what's going on there? What do you think is happening that you have vascular smooth muscle cell progenitors, TRPV1 labeled positive, that look like they have exosomes? So we did a first experiment where we took brown adipose tissue, isolated the fat cells, and treated with one exposure of parathyroid hormone. And what you can see is, and we did males and females, is that particularly in males, there's this marked increase in CD63 positivity in vitro. So we then repeated that using in vivo PTH. So we gave one dose of PTH. And here you can see that, particularly in males, there's this marked increase in CD63 positivity. So that got us very interested in the idea that, one, PTH is working on some progenitor cell line that's not only inducing a thermogenic program, but also may be producing exosomes. So we then looked at the bone marrow of those animals that were treated with PTH to look for CD63 positive GFP within the marrow itself. And we had previously excluded that as being recombined in the marrow. So we knew this was not happening directly in the marrow, the TRPV CD63. And about 5% of the bone marrow cells were labeled with CD63, suggesting that, in fact, the exosomes may be coming from brown fat and going to the bone marrow, which got us very interested because we wanted to then ask, what is the target? Where are those exosomes going? And it looks like they're going to macrophages. F480 positivity was markedly increased in those cells in the bone marrow. We have evidence that they're both macrophages and potentially other fat cells. And then some recent proteomics of the circulation from these TRPV63 cells in the bone marrow. We found at least a couple of candidate proteins that are upregulated that come from brown fat that are in the bone marrow. So it's rather remarkable that you've got a endocrine secreting tissue, brown adipose tissue, which we've only recently begun to appreciate, that's actually targeting the bone marrow. So this is our idea that PTH stimulates the PTH1 receptor. Its primary action is in bone by stimulating the recruitment of new progenitor cells, but it also acts through brown adipose tissue to increase adaptive thermogenesis, and it generates exosomes. So the next big question for us is, why would this happen? Why on earth would we have a calcium regulating hormone that is having an impact on the skeleton? What particular reason? The direct effects of PTH on bone are pretty dramatic. So just to summarize this section, PTH can regulate thermogenic gene program. It induces exosomes and extracellular vesicles that may target in the circulation. Oh, and we also saw those CD63 positive cells in the circulation. So we know that they're coming from brown fat, they're going through the circulation, and they're targeting the bone. So why would our system be designed to develop parathyroid hormone, brown adipose tissue, bone axis? So I went back to fish because fish have the PTH1 receptor, and that was the first evolutionary step. But they also have the PTHR2 receptor, which is very interesting because PTHR2 is activated by PTH2, which is a form of parathyroid hormone, but is slightly different than the classic parathyroid hormone. And it turns out that this is also referred to as TIP39. And this protein has a dose response that is very consistent with what we see with PTH1, and it activates cyclic AMP. So what's interesting about PTH2 or TIP39 that was ignored for a long time was that it's highly expressed in the central nervous system, and particularly in the hypothalamus and the subparaventricular areas. So why is that important? Well, it turns out that Dimitrov and colleagues looked at PTH2, or what we call TIP39, and found that if you give TIP39 to mice, you actually can induce an increase in body temperature. Core body temperature is increased. And TIP39 will protect animals from cold exposure. And so what they showed is that in fish, that TIP39 is a stress response hormone, that it comes out of the hypothalamus, and that it's critical for organ response to particular stress. In this case, it's cold temperatures that induce that PTH2. So that raises this really important possibility, and that is during evolution, we evolved parathyroid hormone as a modulator of calcium balance, well established. But at the same time, it's possible that PTH2, which was in fish, was modified to PTH1 as an alternative temperature regulator. And that by going into a colder environment or going into terrestrial existence, that there was a need for another hormone to help protect body temperature. And so our working hypothesis is that PTH evolved from PTH2 as a secondary form of temperature protection. But more importantly, remember I told you that when you get exposed to the cold, you get this increased adrenergic drive. And that increased adrenergic drive uncouples resorption from formation. But how about if during the cold, PTH stimulates brown adipose tissue to produce exosomes that act on bone cells to protect the bone from that uncoupling? In other words, a built-in system to keep the skeleton modulated during times of acute and chronic cold stress. And that's really where we're at now. The idea that these exosomes are actually important protective devices against prolonged adrenergic exposure of the skeleton, which would lead to bone loss over time. So that's where we are now. We've isolated some of the exosomes. We're trying to understand the cargo, which cargoes are being carried by these exosomes, and how they could affect bone modeling. We're currently in the process. We just submitted a revision of our R21. So we talked a lot about R21. So this was a big shift for us to move into temperature regulation. So we got a 48 percentile first time in, and they wanted us to show that PTH induces energy expenditure, show that it protects body temperature. We've done that, shown that exosomes have some role. We've tried to do that. So it's just submitted today, actually this morning I got the PDF, and we'll see what study section says, but we'll push ahead regardless because we think this is an important physiologic process. And we've added a human component to it because of recent administrative changes. So we now have 20 samples from pre-PTH exposure and 20 samples 11 months later for a treatment regimen in a clinical trial at UCSF. So we should be able to compare the proteomics from human studies with intermittent PTH with what we have currently. So, and then, go back to PTH2 and try to understand the physiologic significance. So I hope that I have given you a story anyways of what it may be another physiologic mechanism. Now, I understand that you guys don't deal with cold that much, but John told me that it was 51 degrees yesterday morning. Your brown fat was activated, and you probably had a little bone resorption. So think about that cold, we're never immune from cold. And if you ever travel to New England, you'll be exposed to that. So this work is funded by a number of different support systems. I want to just point out Zero Lee, my colleague who came from Michigan in Orrin McDougall's lab, was doing great work on bone marrow fat and created a new model where you can conditionally delete substances just in bone marrow fat. Sam Costa is now at the University of Pittsburgh, and Matt, who I mentioned, and Carolyn Shevek, a very talented fourth-year postdoc in my lab. So thanks for listening, and I'm happy to answer questions. You can get rid of it.
Thank you very much, Cliff. That was very insightful lecture, something very, very new and exciting. And you're obviously excited by it. But what else could this be doing that we haven't thought of? I mean, you're affecting brown adipose tissue. Will it have an effect on obesity, glucose, hypertension has been raised because blood pressure changes when you do parathyroidectomy. There've been reports of hypertrophic cardiomyopathy with parathyroid hormone. Hypoparathyroidism. So your comments on what else PTH could be doing, and we've been blaming it all on vitamin D, which is totally useless.
Right, that's right. Vitamin D was just put in there as a mechanism to allow PTH to do its thing. That's how I think of it, like that. Yeah, so no, I think there are model systems that PTH might be, impactful in other tissues as well. I think the hypertension story is an interesting story because there are a lot of notes about this.
Yes.
There's also some evidence that chronic hyperparathyroidism can lead to malignancies later on, so yeah, it really is represents an interesting multifunctional molecule. And I just should point out that the PTH1 receptor is expressed on lots of different tissues. So that gives us an opportunity to access those other potential tissue.
Judd.
Oh. Thank you for that lecture. That was great. But I had a question about, hey, sorry, right here. When you talked about the evolution of this and everything, and so when you look at, I guess, comparative PTH,
Right.
what do you see as far as like endothermic versus exothermic organisms, and even like ones that live in cold versus warm environments?
Right. So we haven't looked at that, but we do know fish, that's an important response element for fish live in colder marine environments. So we do know that, but we have not really extensively studied the other aspects of it. But it's really a good question. What's so remarkable is PTH2 acts and looks just like PTH1. I mean, it stimulates cyclic AMP, activates PKA. It's doing everything we're doing in our bodies with PTH in smaller invertebrates. So yeah, it really does represent an area of further investigation.
Cliff, great
Yeah,
talk.
Jeff, thank you, Jeff.
Along similar lines, I noticed that PTH2 functions through the hypothalamus
Yes.
involved in the temperature response. There's a lot of data showing that body temperature is a component of the host response to infection.
Yes.
And
yes.
I'm wondering if, as you look for new
Great.
functions
Yes.
of PTH2,
Yes. Yes.
that you may also look at that.
That's a great point. Yes, that's great, Judd. I hadn't thought about it in that light, but that's right. There certainly might be an element of it. Yes.
So in hemodialysis patients, and their bone
Yeah.
metabolism is all kinds of messed up.
Right.
But one thing hemodialysis patients are exposed to 3 times a week is a acute temperature change that we usually run them at a little bit below body temperature, 36.5, to
Yes.
help cause vasoconstriction to help keep their body temperature up. But are we
Yes.
also causing changes in their bone mineral metabolism by way of changing their,
Yes.
putting them under thermogenic stress?
So I failed to mention one key point. With cold stress, parathyroid hormone levels go up. So that's the initiating signal. Now, why that is, I don't know, but you're absolutely right. So the temperature goes down. That maybe that's part of the mechanism of sustained hyperparathyroidism, secondary hyperparathyroidism in renal disease. And whether that's responsive because the endogenous levels are higher, maybe they don't respond to this temperature change because their PTH levels are high. It's a great question. I need to look through that a little more, but it's very interesting. Yeah.
All this thing talk, thank you very much. I learn a lot.
Yes.
I know you will talk
Yes.
more
Yes.
than 20 years, and when I
Yes.
outside at Harvard, and that know you in the bone field, really the outstanding work going on. So the PTH has been in the clinical for like for treatment of the osteoporosis for more than 20 years probably.
Yes.
Now, right?
Yeah.
So I just wonder the what happened right now.
So that's a great question. We scratched our heads with the same thing because I had to call investigators and say, well, weight is not listed in the table 1. They give the initial body weight, but nobody actually did measurements of body weight over time. And so we had to go back and we got a few measurements from people at Columbia and at UCSF that suggested there was very modest weight loss, 1.5 kilos mean value. So it wasn't dramatic, but it was out there. But I think what struck me more than not having real convincing data was that nobody actually reported that. Nobody actually even thought that weight could change with that. So in the mouse model of primary hyperparathyroidism, where you have an AAV that's expressing PTH in liver, the changes are dramatic. Now, of course, that's overexpression of PTH, but you see this browning of inguinal adipose tissue. You see brown fat expand. So I think it's there. I don't know if we're giving the right doses or physiologically what those bursts of PTH really represent.
Yeah, the great. And since in the field, you are the pioneer in the field, so you may know more. For example, the potential they have the better medication potential in the clinical trial. Do you know what may have the better medication for osteoporosis?
Well, you know, they PTHRP is also in the clinical space now for treating Timlos for treating osteoporosis, and it capitalizes on the exact same mechanism that PTH does. And yeah, there's more anabolics out there, but it takes time to develop.
Okay, thank you.
Yeah.
Really great to see you in the New Orleans.
Thank you.
A quick question. Sorry. You mentioned that the importance of PTH in the bone and the fat content in the bone marrow metabolism, but as you know, the other component of the bone marrow is the hematopoiesis.
Right.
So I want your opinion about that, and specifically, if you think that PTH fluctuations in patients could stress the marrow to the point that could lead into problems such as aplasia, myelodysplasia, leukemia, etc.
It's a great question. So Hank Cronenberg and his group have shown that PTH can be used to help mobilize skeletal stem cells and hematopoietic stem cells to increase white counts and improve responses to different stresses. So the theory was that PTH improved the niche around hematopoietic stem cells to allow them to be more responsive to either grafting or other potential stimuli. I think some of the PTH effect is due to the liberation of fatty acids that are utilized by hematopoietic stem cells to get them going. So I think it's a positive effect, and I think it hasn't been shown to cause marrow aplasia or dysplasia, but you do lose some of the marrow fat component. And we know from just stem cell grafts that after radiation, the first thing that appears in the marrow is fat tissue. And it's once that fat tissue begins to decrease that you see these hematopoietic stem cells coming out, suggesting that the fat is there to help those stem cells really be energized. So that's our thinking about it. Not sure if you're...
Thank you. As you went through your slides, all I could think of is estrogen is involved in everything.
Right.
And sex differences, really, I shouldn't say estrogen.
Yeah.
So my question is, have you looked at sex differences? And I think it would be interesting to see sort of normal estrogen levels compared to postmenopause or whatnot.
That's a great question. And we have, so interesting, and I went over a little too quickly, but male black 6 mice respond better to PTH at the skeletal level in terms of their change in bone density is greater in males than it is in females. And we found that with the exosomes and the induction of thermogenesis, males were more responsive as well. So we're trying to understand what estrogen is doing in that space. So in anorexia, we did a study with Ann Klebansky, Miriam Bradella, where they gave parathyroid hormone to anorexic women who had very low bone density. And most people were very anxious about that, but it turns out that the skeletal response in anorexic women was twice as great as it was in normal women. Now, they don't have estrogen, which is interesting because they're amenorrheic, and they have tons of marrow fat. So I think the response was due to the extra energy that was available for those osteoblasts. But, you know, sex differences are such an important component, and nobody understands it. Nobody can really, you know, we get these reviews back from studies that show, oh, yeah, you know, how can you show that males and females are different? What's the mechanism? Estrogen has a lot to do with it. Estrogen also downregulates marrow fat. So if you look at postmenopausal women without estrogen versus those that have been replaced, the marrow fat differences are amazing. So estrogen prevents that marrow adiposity. And that's one of the best ways to induce marrow fat experimentally in female mice is just do ovariectomy.
Thank you.
All right.
Thank you very
Okay.
much.
Okay.